Okay, hello and welcome back to this lecture 20 on bio microelectromechanical systems. We'll just do a quick review of what happened in the last lecture. So we discussed about capillary electrophoresis, which is essentially the fractionation of DNA molecules through thin capillaries loaded with gel materials, okay, agarose, hydroxyalkyl cellulose, polyacrylamide, so on and so forth. So the advantage here is that essentially the detection throughput of these microcapillaries is much more than the conventional gels. One of the reason being that because of the high surface area to volume ratio, the temperature of these gels, the amount of heat that these gels can absorb without getting broken or molten is really very high, and therefore it can withstand higher amount of electric fields or higher amount of EMF, electromotive force. So voltages as high as about 1000 to 3000 volts can be successfully applied into these microcapillaries in order to move DNA fragments. And therefore essentially there are twofold advantages. One is the increased mobility because of the higher electric field. Another is the ability, because this is a microcapillary, it is overall lesser in size. You can accommodate a dense network of such capillaries in a very small area and can do effectively an array based detection. So it's a high throughput system which you can generate using capillary electrophoresis. We also talked about space domain PCR reactors and how to design them. I'll just like to reiterate again that all PCR microchip devices have been categorized into time and space domain devices respectively. The time domain device, there is a small chamber, and essentially this chamber is circulated through 3 or 4 zones of temperature, 3 or 4 different points of temperature, as is true for a normal PCR process. And there is a general confinement of the volume, the reaction volume inside that system. And in the space domain devices, you have to kind of oscillate physically this PCR fluid as a droplet or as a micro droplet over the 3 different heating zones, okay, and this lets you or helps you to avoid the ramp up ramp down additional time as the time domain device would require. So spatial designs, spatial domain space PCR reactors have been designed, and essentially what happens in these cases is you have to design it for the maximum velocity so that you can do the robustness of design. Even though the fluid is moving at its peak, the PCR fluid has enough interaction time with the temperature zones for it to get denatured, annealed and extended according to whatever process step it is in. And essentially space domain PCR comprises of a channel which is engraved over these 3 differential heat zones and then given multiple turns. So we designed such a reactor device. We also talked about a little bit about fabrication processes for fabrication of these capillaries, especially for electrophoresis platforms. Then we did understand DNA mediated self assembly by referring to one of the very first works in this area from Alvesato's group at Penn State. And here it was one of the pioneering works to start this whole field of DNA detection through DNA hybridization. We also did some basics of genomic detection, how hybridization can be used as an identification for detecting DNA molecules, and then finally went to this very fascinating, very new area of microarrays, gene microarrays. So we'll kind of start from this area today and do some theoretical studies about the different commercially available microarray systems. As on date, there are about 2 companies in the world which produce these arrays from 2 different approaches. One is Alphametrics, another is Nanogen. Their approaches are totally different for making or realizing the capture probes on a surface to create the gene library, okay. And these cards which are immobilized with this library are either sold in one of the approaches directly to the customer, and another approach, it is basically the card platform which is sold, and the customer is expected to immobilize himself the particular capture probe in the particular area that he intends to do. So let us look at what hybridization really is and how microarrays function. So you have these capture probes, as you can see here in this particular slide, of multiple DNA molecules, okay, and these capture probes are immobilized onto this surface at the bottom. So more so like this library. So you have S1 all the way to S9, S1 through S9, different capture probe sequences, and somehow if you can direct sequence S1 to this square in the first row and first column, S2 in the first row second column, and so on and so forth, you could actually make or build a library of these sequences in these different boxes, as you can see here, okay. The advantage is that when you actually have a target DNA, and this right here is a target DNA, and are able to label it using a fluorophore molecule, and the target DNA has the exact complementary sequence of the capture probe that is there on the surface here, then this target DNA kind of binds to the capture probe, and then when you wash the whole sample, the unbounded sample is removed except the target DNA which is bound, okay. And because of this binding, and also because it has already been conjugated with a fluorophore, you can see in one of such spots or one of such areas there is a growth in fluorescence. So if there is a growth in fluorescence, that means there is a perfect complementary probe which is present in this particular area corresponding to the target sequence. And if you already have an information of the library as to what capture probe was there in that particular area, you could gauge the sequence on the target DNA molecule. So essentially this is also the basis of detection of nucleotides, okay, this hybridization principle, and it's basically the hybridization of an unknown fluorescently tagged strand with many known strands, and these are DNA strands. The reaction will determine the sequence of the unknown or vice versa. So the question is, how do you really make these particular strands here, as you can see, with a certain sequence which is exactly complementary to some of the targets that you would be planning to pick up eventually, and how do you actually make these molecules in these different boxes or zones, as is illustrated by this this big square. So there are two approaches really which can be used for doing these kind of studies. One is where the strand can be lithographically or placed on the surface. That means you can actually put the DNA strand with the light directed approach or light direct synthesis that we are going to look in just about a little bit, or alternately you can actually direct the strand to the particular position by using an electronic addressing system, as is normally done by this company Nanogen, defined at a specific location. Okay, so that's how really the concept of hybridization works in biochips, and let us actually look at both these strategies independently. How do you electronically place the DNA probes, as is the approach used by Nanogen. So here in this particular instance, the company really supplies the platform, the microscale platform, which is actually having this electronic addressing system for directing a particular capture probe of interest to a particular area on its surface, okay. And the vendor or the manufacturer really supplies this platform without any immobilization to the customer, and the customer, who is a biologist, is expected, or a bioengineer is expected to kind of put probes in a certain sequence by using this platform and electronically addressing independent pixels, as I'll show in just about a little bit how this addressing can be done. And the customer actually takes care of building his own library according to his own requirement, okay. So this is the basic difference in the Nanogen and the Alphametric approach, that the Nanogen is a company which would provide you the platform, and it would provide you the protocol for immobilizing the capture probes, and it is essentially your job as a customer to immobilize the capture probes that you are looking at and create your own library. So there is greater flexibility that such a product would offer, as opposed to the other approaches where they already build in the library and give it to you at the very outset. Okay. So here if you see the Alphametrics, the Nanogen approach, really you have this wafer as you can see here, and you also have these metal contacts. Okay. And these are connected through conduits to a basic circuitry, and the connectivity is in a manner that you could provide a charged surface you could provide a charged surface onto the electrode. Okay. So that's how you have connected the particular electrode. So let's make this electrode connect to a positive DC voltage source terminal. So what do you expect will happen? So there would be a tendency of these capture probes, and now so you place this particular assembly into a solution which has the capture probe for type 1. So it has type 1 capture probes, the solution above it. And so when you are actually applying a positive voltage in this electrode, it is very natural to assume that the DNA would move towards the capture probe using electrophoresis. Okay. And the capture probe slowly kind of comes and settles down on this particular electrode, because this is the only one which is charged positively. Okay. So there is actually a lysine layer here in this area which can do this job of binding electrostatically the negative DNA onto the surface. So you have a layer which would electrostatically bind the negative DNA once it has approached this particular electrode onto the surface. Now if you want to put another library or another probe type on, say, let's say electrode 2, okay, this is electrode 1. So in that case, if you apply a voltage, a positive voltage in electrode 2 here, there would be a tendency of the capture probes, and remove the voltage on the first one. Okay, you have to remove the voltage essentially on the first one. So the capture probes now would like to go towards this middle electrode here, and the polyl lysine film here would again kind of stick to the capture probe and immobilize it. So there is again a second set of capture probes which are put now in this over second electrode. So you have one capture probe in the previous step and one in the next step, and so therefore you can repeat this process many times. So all the different electrodes has a set of capture probes, and mind you, in this case, as we know what addressing is being done, and if we change solution in between, we have a very well idea of what is the sequence on the capture probe that we are putting on a particular electrode. So essentially we have the information for the whole library of the capture probes, which are this way immobilized over the whole silicon wafer with the electrodes. Okay. Now we can actually pull the target DNA in pretty much similar way, but prior to that we also have to ascertain that they have some kind of an optical transduction mechanism which can make them detectable. So basically the target that you put inside the solution now, a clean, let's say a DI water sample maybe. So the target that you put there, the target DNA, should essentially be labeled, and it should be single stranded structure that you would like to monitor on the sequence. So now when you can actually put this green fluorophore labeled single stranded DNA molecule as you are seeing here, and now start applying a potential difference. Okay. So you are starting to apply a positive potential now in these electrodes. So there is a tendency of these molecules to kind of come close. Okay. And these would now based on whichever one has the potential, let's say in the first instance only the middle electrode has been given or provided a potential. So they would all kind of emerge into this middle electrode and try to bind on the DNA which is there on the middle electrode. Effectively what it would mean, and this is shown here, so essentially you have electrophoretic mobility again driving this negatively charged DNA onto the DNA immobilized on the middle electrode. And so mind you, these are all fluorophore leveled, and if suppose it is complementary to the capture probe, or we are going to test whether it is going to be complementary to a particular type of capture probe or not, and if suppose it is positive in that sense, there would be a binding action. And so when you wash this particular chip after the electrophoresis process is over, it does not get rid of these particular fluorophore labels. So therefore there is a pixel which is essentially turned on, from which one can gauge what is the sequence of the target that you are looking at. Now you could also use the electrode for a concept of repelling the negatively charged single strand. So suppose now you have already registered a complementary binding over this particular electrode, and this is now futile, you cannot use this electrode anymore. So what you do is, in order to prevent any further binding of the negative of the other single stranded DNA, so that the target concentration is not lost in the next cycle, you apply a small negative bias here, so that nothing comes near this particular electrode once it has been already bound and the molecules stay back, right. So this negative bias is used, and the other two can then be converted into a positive bias, so that these molecules can drift essentially towards both the sides, okay, and then start binding into this immobilized DNA molecule. So this is how the nanogen approach is essentially developed for making the capture probe. It's a very simple process. In a laboratory also, the experimentalist can actually go ahead and make a library of his own choice, and then can use that for identification of certain target DNAs that he is looking into his samples. All right. So the summarily speaking, the DNA biochips from Nanogen has the following features, okay, and this actually has been borrowed from this website here, this picture, www.nanogen.com. Similarly, this particular example has been borrowed from this cited reference here. It's one of my own earlier papers doing review for these kind of systems. And so essentially in the Nanogen approach, the chief technology features are the following: biochips for DNA detection, antigen antibody, enzyme substrate, cell receptor, cell separation techniques. All these kind of chips are available. It takes advantages of charges on the DNA or any other biological molecules if you are detecting proteins or antibody, antigen, etc. Every time it uses the advantage that some of these molecules are charged, or it has a huge charge at least in its backbone, and then small sequences of DNA capture probes are electronically placed or addressed to specific sites on the microchip, which can be used for grabbing now more and more different target DNAs. So this is how the chips really would look like once the fluorophore labeled target DNA is immobilized, okay. So here is probably one corresponding to this yellow fluorescence, another red fluorescence has come up when the red fluorescent labeled DNA, single stranded DNA, binds to the capture probe which is on the surface. Similarly, you have an array of these molecules. This is the pink fluorescence, this is probably the blue or the green fluorescence, and so depending on what is the particular capture probe or what is the sequence on the library of the capture probe, it captures different targets, and we can immobilize, or we can actually know where it has been captured by using different fluorescent labels on these multiple targets, so that you can have rows or columns of one colored fluorescence, which gives an idea of what really the sequence on the target DNA is. We very well know again the sequence on the capture probe, because we are the one as an experimentalist who are actually designing the library and making the library on the top of the chip. Okay, so these are some of the main features of the Nanogen biochip. So essentially what we can say is that in a nutshell, the hybridization mechanism is the primary detection mechanism used in these biochips, wherein a test sample can be analyzed for the presence of target DNA molecules by determining which of the DNA capture probes on the array bind or hybridize with the complementary DNA in the test sample. So if there are some rows or columns which are left untouched and there is no fluorescence growth or no pixel turning on, that means those capture probes are certainly not having the complementary sequence of the target. But when the moment there is a residual fluorescence after the wash cycle is over, it automatically means that corresponding to whatever was there in the library part or in the part of the library, and if we are able to know that sequence, we can really predict what was there in the target as a DNA sequence. So these are some of the commercially available protocols. As you can see, this is essentially a reader card mechanism. Okay, so essentially this is the main mother microchip, okay, and the housings surrounding is really the reader mechanism. And so these are very available, very compact manner nowadays in order to do hybridization based array detection of DNA. If you look at the back on the back side for the electrical connectivities of this particular chip, you can find out very easily how it looks like. So essentially these are all the different contacts that you provide in order to provide the electrical voltage to the different array of pillars or posts of principally gold, these pillar or posts, as you can see here, which are carved or manufactured on the top of the chip. Okay, so this essentially is the reverse circuitry of this particular chip here, which gives you the connections between the various the electrodes on the surface, and you can actually keep biasing them positive or negative depending on your requirement. Okay, so there is another approach which this company Alphametrics follows. So in this approach, basically a beam of light is used to build capture probes molecule by molecule. The way you do it is that you take a wafer, okay, and by the way, this actually this technique was reported by Fodor et al. Back in 1991, which started this company Alphametrics. Okay, it's a very innovative approach to build molecules. So you take a wafer, and let's suppose these are different photomolecule capped linker layers. Okay, so there are two aspects in this particular layer. One is that these blue dots here, as you see, are photomolecules, okay, and they are capped over this linker layer, which essentially bonds the photomolecule to the substrate. So in this particular approach, the first step is that some of these capping layers are first removed, they are uncapped, okay, and the way you do it is by exposure to light. So you have this light source here, and you do a light deprotection by using a mask. We have been talking off and on about what a mask is. It is essentially a kind of black and white transparency, or a chrome coated hard substrate made up of glass with certain features and designs which would be transparent. So wherever there is a transparent via or structure, light kind of goes into and decaps or uncaps these photomolecule layers. Okay, so you have deprotected this linker layer here by using a light signal. So you are deprotecting the linker layer in this particular illustration. So once you do that, you actually replace this linker, one of this top linker layer by CH, okay, groups, and use these particular groups to replace the thymine molecule, which has this capping layer at one of its ends. So you have essentially uncapped these linker layers, replace them with CH groups here using a light deprotection technique and masking step, and then you in the solution you do have these different adenine, thymine, cytosine and guanine, each with these photomolecules, a capping layer. So you take, let's say, so you have to grow the thymine in these two sites, site 1 and 2, let's say that is the plan. So what you do is you actually use the thymine coated or thymine conjugated molecular or this thymine conjugated capping layer to this particular molecule, such that you replace the CH here by the thymine group, okay, and the arrangement actually does a capping back of the opened pixels in this particular step. Okay. So, once this is done again, you can use again a different mask with an exposure in two other areas or two other sites to open the linker layer again by decapping these two capping molecules or capped molecules or photomolecules, and then you are again doing the same chemistry wherein you have this OH bonded to this area, and then maybe in this case you are applying a cytosine coated with a photomolecule, so that it comes and bonds to these two sites here which have been opened up, and after bonding it again gets covered with this small capping molecule. Okay. So, essentially you are doing a series of bonding and capping simultaneously, and when you keep on opening and closing such capping molecular layers and keep on building the whole library, this right here illustrates a 25 mer library. Okay. So, you have the C, A, T so on up to G 25 in number. So, it is like a 25 oligomer library that you have built over each of these pixels in a different sense or in a different sequence. So, this essentially is again a series of masking and opening of the photomolecular layer and conjugation with a certain group, which again brings in a photomolecular layer, and then doing the masking process again to open another site. So, you are building a library, essentially planning your opening and closing of the photomolecules accordingly. Okay. So, that is what the light directed synthesis approach of Alphametrics is. In this particular case though, as in contrary to the Nanogen approach, the Alphametrics vendor actually supplies with these chips which have been developed in their laboratory, and they do have spec sheets which they provide with the particular microchips, which talks about the library and the capture probe information in different areas of the particular chip. Okay. So, this is again a very different approach than the Nanogen approach. Here the flexibility of using your own capture probe may be limited, because the vendor would give you this particular capture sequence or a capture probe already synthesized at their end. In a sense, what you can do with this synthesis is that you can actually try to build a dense combinations of chemical steps, or integrate a dense combinations of chemical steps to build really long capture probe arrays. Okay. So, here as you are seeing in round 1, you are essentially opening and closing the different layers using masking process, so that first the A gets sutued, then the G gets sutued, then C and T. So, there are 4 steps essentially here. In round 2, you may actually decide to go horizontal, and then try to make a crisscross arrangement, so that you have now a combination of let us say an A with an A here, a G with an A here, a C with an A here, or a T with an A here, right. So, this is layer 1, this one is layer 2. Okay. In the third step or round 3, you can do a layer 3 from this end, okay, and so on so forth. Then in round 4, you could again do the layer from this particular end, which is dotted here. So, what I would like to just kind of assert here is that the various layers of a certain capture probe could be just drawn by rotating the direction of using or the direction of placing the mask with respect to the wafer surface, and that way you could have a dense integration of all these different molecular layers one over the other. So table 1 here really talks about, and this has been again borrowed from this paper Fodor et al. And it's also listed in this Alphametrics website essentially. So the combinatorial synthesis of polynucleotide probe arrays would essentially mean that so many probe lengths, let us say length of 4, would essentially take about 16 chemical steps, and the number of possible combinations that are available is 16 times of 16. Okay, so it is 256. So essentially what it means is that when you are actually having a probe length of 4, all right, so you are trying to build a sequence, let us say A, T, G and C, 4 probes. So for these 4 probes you have to perform 16 steps, okay, as you can see here. So the first step can be from this end, all right, let's give me a minute here. So the first step could be from this end, the second could be from this end, the third could be from this end, and fourth could be from this particular end. So you are essentially rotating the way that you can align the mask with respect to the wafer, and you are essentially going through 16 steps in order to get the first layer, the second layer, the third layer and finally the fourth layer, okay, of the molecule. So it can be, so if you want to build like an array with 4 different layers all to the surface, you need 16 chemical steps for this 4 layer. And the number of combinations that you can have is essentially if you assume 16 possibilities, okay, in 16 chemical steps you have about 256 number of possible probes. So similarly, if the probe length is 8, the chemical steps is 32, the number of possible probes is 65,536. And so you can see here that the way that this combination of different sequences is increasing is tremendous. It can go up to almost 10 to the power 12 with a probe length of only 20, which would involve about essentially 80 chemical steps to get realized, okay. And therefore it is really of vast utility. The library can be very diverse, and also it can develop a large number of possibilities just with a few probe lengths on the particular surface. So light directed synthesis in a way is a very simplistic way of understanding how you could capture different combinations, okay, all together on a single surface, different molecules. So all these gene chips, whether it is Alphametrics or Nanogen, essentially would look in the product form something like this, all right. This is essentially the readout of such a chip. If you can see here, there are these fluorescent pixels which have opened and closed at various regions of this particular chip, which essentially signifies the binding of the target DNA molecule, and therefore the capture probe which may have been in this region would have corresponded to the target. Similarly, in this particular region would have corresponded to the target, and you can get an idea of how what would be the sequence of the capture probe if you could already know what is the existing capture probe which is here, okay. So you could get an idea of the target the target molecule if you can know, or if you can have prior information of what is there as a sequence of the capture probe. So that is how a real Alphametrics readout would really look like. So ultimately fluorescence detection is the basic means of finding out whether binding has happened or not, and ultimately this fluorescence detection would be able to limit the size of the pixel in the array, because you cannot really go on synthesizing smaller and smaller just because of the fact that you have to have enough fluorescence intensity for the detector to be able to tell you independently whether it is a pixel or which has opened up, or which signifies the presence of the fluorophore. And so if you go on reducing the size, there may be a drastic loss in intensity and the detector may no longer be able to sense. So that is a limitation point for designing the pixel size, the final pixel size, although using fabrication you could have probably taken it much lesser in term almost orders of magnitude less than the existing size. So the detection imposes the limit on the final size of the array, okay. And these are immensely utilized nowadays, these different so called gene chips using various approaches in various laboratory related experiments. Polynucleotide array, HIV resequencing, mRNA expression monitoring, some of these protocols are very often in great need of such gene chips to tell you accurately, rapidly, quantifiably how much amount of what can be the sequence on the target or what can be the concentration of the target. So one more information that you get out of this fluorescence readout is the fluorescence intensity, okay. And the fluorescence intensity can signify what is really the binding level in a particular capture probe. And so if the intensity crosses a certain threshold only, you take the data in that case, and also beyond that threshold, whatever change in intensity is recorded is also a change in concentration of the target molecule. So therefore the fluorescence intensity provides, in addition to the yes no type answer of the sequence, an idea of what could be the concentration of the particular target sequence. So that is exactly what these gene chips essentially do for gene analysis. So there are other protocols and means for detection of certain sequences of DNA. This one approach here that we would discuss next is essentially a chip which has been developed by Motorola, and this was reported also in this Nature Biotechnology journal back in 2003. And here this is what the chip really looks like. If you look at the whole platform, this is what the gene chip really is. And here the detection again is based on a label which is more like an electrochemical label, okay. So the capture probes are attached to the electrodes in the first step, and this here, if you look at, is really the capture probe which has been bound. This blue stain here is the capture probe which has been bound all the way down to this alkane linker, okay. So the substrate preparation in this particular process is very critical. The substrate is prepared in a manner so that you have molecular wires. So in this this particular illustration here, as you can see, there are, the surface is not really a plain surface, it is pretty complicated. There is a gold electrode surface which is shown by the blue here, okay. So this is essentially the gold electrode surface. There are alkane linkers which are in this particular area as an insulator, okay. There is a molecular wire which is there inside these alkane linkers. So the molecular wires essentially are the conductors, okay. So there is an alkane linker which is an insulator, and in parallel to it there is a molecular wire which is a conductor. So it conducts, it's like a a nanoscale conductor which would be able to detect a single electron transfer event. So the way that this works is based on electrochemical sensing, and if you can see here the signaling probe, okay, this essentially is the signaling probe. Sorry, this let's just rub all this off to make this thing a little more clearer. So this essentially is the signaling probe. This other end, this complementary end of the DNA, it has a signaling. Why it is called a signaling probe is because it varies from 3' to 5' side of the DNA, and it contains an electrochemical label called a ferrocene group. Okay. So, what a ferrocene label typically would do is it essentially is an electron transfer agent. It transfers electrons from the ferrocene label when in close proximity to an electrode. Okay. In this particular case, the label, if you can see, is bonded molecularly to the signaling molecule like this. All right. So, you have a capture probe here bonded to one of these alkane linkers, which is an insulator, and you have a signaling probe here which has a ferrocene group and which is lying somewhere around in the surface. So, this is the situation. Now, let us suppose we have a target nucleic acid which we want to detect, okay, and very intelligently the system can pick this up by aligning the signaling probe flat to the bed of the molecular nanowires and getting the ferrocene very close to the molecular nanowires. Let us see how. So, you have a certain sequence in the capture probe and a certain other sequence in the signaling probe. Now, these sequences can be altered in a manner that a large DNA molecule, let us say a large single stranded DNA molecule can bind to both the capture probe as well as the signaling probe. So, let us suppose this 3' to 5' molecule here is the target nucleic acid, it is a single stranded DNA molecule, and it has groups complementary to a portion on the capture probe, okay, and also another portion on the signaling probe. So, essentially it is making the capture probe and the signaling probe kind of one integral by binding halfway through the capture probe and halfway through the signaling probe. But there is another effect which it leads to or generates, you know, when this kind of a binding happens, there is almost always a perpendicular angle between the signaling probe direction and the capture probe direction. Okay. So, when the binding event happens, the binding event kind of ensures that these things are just perpendicular like this. So, this is the capture probe standing upright and this is the signaling probe and they are just perpendicular to each other. So, it gives some pressure to the ferrocene molecules which are just simply lying on this wire of brushes which has an alkane linker which is essentially an insulator in nature. So, as the pressure is given because of this perpendicular alignment suddenly of the capture and signaling probe, the ferrocene label kind of gets to a little bit depth, crosses the alkyne linker layer and comes very near these molecular nanowires which are a little shorter than the alkyne linker, and therefore, the single electron transfer events are rapidly recorded. So, as they are recorded, it gives an idea of whether the DNA has bound both to the capture and a signaling probe. Now, this is a little complicated process, and essentially there are a few reasons why this kind of a technique or technology works very well. One is the specificity of detection. So, if you think about a case when there is a DNA molecule which has a sequence which is aligning not only to a capture probe, but also to a signaling probe, to consider this electron transfer effect or event to happen, it is a much better selectivity over just a normal hybridization assay which has only one capture probe binding to the target DNA molecules. Okay. So, that is one aspect of this. Another is that electronic labels, especially electrochemical labels, have always been the buzzword in the industry because of the fact that it can align them very well with microprocessors, so that they can do automatic measurements, data analysis, signal acquisition, so on and so forth. So, therefore, microchip architectures almost always have been associated with some kind of a electrochemical or electrical detection. Okay. And so, this thought process of the signaling probe also falls in line with the same idea. So, because of these two reasons, this is a very well known architecture which is currently available for DNA detection. Motorola is a company which actually sells some of these chips for laboratory usage. Okay. So, essentially, in a summary, there is a target DNA which binds to complementary probes. Okay. the capture probes are attached to the electrodes. DNA sequence is called signaling probes with electronic labels attached to them. Okay. So, ferrocene modified DNA oligonucleotides E1/2 or E half, the EMF generated in this particular example is about 0.120 volts, and this essentially ferrocene modified DNA essentially acts as a signaling molecule by emitting an electron, and binding of the target sequence to both the capture probe and the signaling probe connects the electronic label to the molecular nanowire which is there on the surface and which can do the electron conduction, and the labels transfer electrons to the electrode surface producing a characteristic signal which would give an indication whether the DNA that you are looking at, the target DNA, has base pairs complementary to the capture as well as the signaling probes. Okay. So, in a nutshell, these are some of the DNA hybridization arrays which are as on date available commercially in the market. So, I have been probably able to kind of take you through at least some of the few integrated gene chip arrangements that are commercially available. Now, let us look into a kind of totally different aspect of how to sequence a DNA, and this reaction is also popularly known as the Sanger's reaction, developed by a scientist called William Sanger in 1975, for which he subsequently won the Nobel Prize. It is one of the first steps in identifying the sequence of information that is there on a DNA molecule, and this is also very foundational for the field of molecular biology or diagnostics to have developed. It kind of emerged from this sequencing activity. So, let us look at how this reaction has been designed in a step by step manner. So, to do this thing, first what we need to ascertain or find out is what all are the constituents of this reaction. So, let us say we are trying to sequence this particular DNA product which is from 5 prime to 3 prime, it is a single stranded DNA structure, and let us say you want to find out what follows what in terms of these molecules. Okay. So, as you are seeing here, this molecules A, T, C, T, G, 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 C and so on so forth is the sequence of information that is really not visible at the outset when you have this DNA molecule with you. You want to find out what is there in the sequence looking at this molecule as a whole. So, this reaction is also popularly known as the dideoxy chain termination reaction. Okay. Dideoxy chain termination reaction used for gene sequencing. So, let us suppose as we were talking before, we want to detect the sequence on this particular molecule, the single stranded DNA molecule which you are seeing here from 5 prime to 3 prime. So, you need the following for detection. You need a DNA polymerase. You now already know how the DNA polymerase works. It is an enzyme which kind of zips the DNA open and then tries to suture the 2 daughter strands to make complete strands by fixing oligonucleotides from the solution one by one on the 5 to 3 prime direction, and from the on the 3 to 5 prime direction it does so by producing fragments, the Okazaki fragments which we discussed in about the last lecture. Okay. So, that is what the DNA polymerase is. So, you need DNA polymerase for this as well. You need the nucleotides A, T, C and G independently. It is called DNTPs. So, you have 4 different DNTPs, and then you have a dideoxy chain terminated, let us suppose ATP in this particular case. Okay. So, it is a dideoxy chain terminated nucleotide, also represented as this here. This is the chemical structure of this nucleotide. So, the advantage of the dideoxy chain terminated nucleotide in such a reaction is that as and when this particular nucleotide binds to a certain region of the chain, the reaction terminates. That is why it is called chain terminating nucleotide. So, whenever this dideoxy, the dideoxy group essentially prevents the further development or suturement of the sugar molecule to the next phosphodiester linkage. It gives an energetically least favorable configuration, and so therefore, whenever there is a dideoxy group which comes and sets towards the end of the chain does not replicate anymore, it kind of terminates there. So, we use this as an idea for stopping the reaction in a controlled manner as and when we require it to stop. Now, also important to mention here is that this dideoxy group has some kind of so what is important for me to tell you is that, particularly for this dideoxy group in this particular case, it has a certain concentration with respect to the DNTP. Okay. So, the dideoxy chain or the dideoxyl nucleotide is of a certain ratio in comparison to the actual DNTPs which are present without the dideoxy group. So, normally in all Sanger's reaction processes, the DDNTPs, as we popularly know them, the dideoxy NTTP is about 1% by volume of the DNTPs. This is a standard which is followed in the reaction design. Okay. Now, interestingly, when this kind of a reaction proceeds, okay, and let us suppose we have a DDATP as in this particular case, the DDATP. So, there would be fragments produced from this DNA wherever there is a binder molecule T. We know that the thymine binds to the adenine, okay, from earlier knowledge about the DNA. So, whenever there is a thymine molecule somewhere placed in the chain, the adenine molecule, I am sorry, somewhere placed in the chain, that may be a point of cleavage or cleaving the molecule as and when the dideoxy group binds with the thymine on it on the particular A. So, suppose there are 50 centers with the nucleotide A on the parent strand of the DNA molecule, and this dideoxy chain terminating thymine group, which is present as 1% of the normal DNTPs, okay, that kind of binds, whenever it binds to any of those 50 sites for A, it terminates the chain. So, the end result of this process or this reaction is a set of fragments of chains which are terminated each at the adenine. Okay. So, it kind of gives a location to us of the adenine on the particular base pair, on the particular parent strand of the DNA molecule. Now, let us suppose we have 4 different such reactions with dideoxy ATP, dideoxy TTP, dideoxy CTP and dideoxy GTP. Each of them are 1% of the normal DNTPs in the reaction, and these 4 reactions are all independent, okay, in 4 different vials. So, what is going to happen is that wherever there is the presence of a G molecule would be terminated and fragments would be developed in the reaction vessel which contains the dideoxy G molecule. Okay. Similarly, when there is a fragment present of let us say the adenine, when there is an end group or a group that we are trying to decipher is guanine, in that case we can produce the fragments of wherever the guanine was present in the parental chain by putting it with 1% DCTP or dideoxy CTP, cytosine nucleotide. So, if we take all these 4 products and run it on a gel, okay, something like this. So, what happens is that wherever there is a chain termination on the ATP would develop based on sizes these fragments which we can later on blow up by using fluorescence. For the DTTP case, there can be fragments in this manner, and similarly for the CTP and GTP. And if we do an overall readout, we will be able to get the position of what follows what in this particular chain. So, I am going to kind of take this ahead a little bit later. We have almost come towards the end of this particular lecture. So, the next lecture we will talk about the Sanger's process a little bit more in details and try to see how micro scale architecture can be kind of synergistically developed with respect to Sanger's reaction to do rapid sequencing. Thank you.